Welcome to the Fungisphere webinar series, brought to you by the Sydney Infectious Disease Institute here at the University of Sydney. I'm Associate Professor Justin Beardsley, and it's my pleasure to introduce these talks, our passionate team, and share some of our mission with you. We're dedicated to increasing our understanding of all things fungal, be that human health, animal health, agriculture, ecology, or even the productive and industrial use of fungi. We are seeking to uncover the threats and the promises offered by these remarkable organisms. This webinar series is more than just a series of talks. It's a platform to bring experts in the field together to share their knowledge and hopefully to build new connections. It's a testament to the synergies that exist between researchers here, but also colleagues globally. Through these talks, we share some of the incredible work being done here in the University of Sydney, but also provide a platform for international experts to share their groundbreaking research too. So, whether you're a seasoned researcher, a student, or simply somebody with an interest in the fascinating world of fungi, you'll find something of interest in this webinar series. Together, we're going to explore the threats and the promises of the fungal world. For today's talk, we welcome Dr. Meghann Thai. She's an expert on the microbial communities that underpin button mushroom production. She uses a combination of phenotypic and molecular methods to understand these communities in order to maximize productivity. So let's hear from Meghann on this fascinating topic.
Thank you for the introduction, Justin. And my name is Meghann. For those of you that I met last week at the Fungisphere event, it was fantastic to meet you. And I'm super excited to meet everybody else as we go through. So today I'll be talking to you about how to grow mushrooms. Mushrooms come in all sorts of shapes, colors, and sizes. They are known for being great nutritional value, particularly being high in protein. And in Australia, they are the sixth most important horticultural crop, which 90% of that is taken up by the button mushroom industry, which I'll be mostly focusing on today. So how do we grow button mushrooms? Button mushrooms are grown on a composted substrate made from wheat straw, chicken manure, and gypsum. The raw materials are piled together and placed onto a concrete slab where it's wetted and allowed to partially ferment for about 9 to 21 days. This process is mostly aerobic. The compost is regularly turned. And also some compost yards do add some air throughout the process. However, the temperature profile is largely uncontrolled. So this starts off initially mesophilic and then rapidly becomes thermophilic, usually within about 24 to 48 hours. Following that, the compost has now been partially fermented, is now moved into temperature controlled tunnels where it's completely sealed. This process is highly aerobic, where air is forced through the entire compost profile at a very fast rate, and it undergoes two stages. So the first stage being pasteurization, where we inactivate a lot of the pathogenic organisms, and then a curing conditioning phase, where the compost is then cooled down to about 45 degrees. So this process takes about 6 days, and this is quite a static process across all of the compost yards in Australia. After that, the compost is then inoculated with mushroom spawn, which is the agaricus mushroom mycelium that's grown on grain. That's inoculated into the compost and allowed to proliferate throughout the compost. And this takes about 16 days. Also highly aerobic as well, so it's in the same sealed tunnels that phase 2 occurs in, and it will grow throughout the compost. Then the compost is then transported to farms where they will load it onto these shelves in large rooms. They'll add a low nutrient peat layer that we call casing. And then after about 19 days, we have our first harvest of mushrooms. What's happening in the compost? microbially. The heat that's generated by the microbes during phase one is due to the microbes using the carbon and nitrogen that's already there and easy to use. A lot of ammonia is released from that due to the heat they're generating, as well as from proteolysis. Then once it's moved into those sealed tunnels, that heat and the ammonia aids in killing the pathogenic organisms. And then during that curing stage, where the rest of our organisms that are there will incorporate that free ammonia back into the compost as biomass. Then in phase three, where our mushrooms are growing and proliferating throughout our compost, they get their nitrogen from that microbial biomass, as well as getting carbon by releasing lignin degrading enzymes. So what are the fungi, what are the microbes that are in the compost? So fungal succession, we get this rapid succession of fungal taxa at the start. And then in phase two, we have a complete dominance by Mycothermus thermophilus until the compost is then inoculated with the agaricus, where it eventually takes off and the Mycothermus eventually dies down. So Mycothermus has been extensively studied since 1992 for its growth promoting role with Agaricus bisporus. This particular organism is one of the key organisms for that ammonia incorporation that occurs during phase two. And it also produces cellulose and hemicellulose degrading enzymes, breaking down a lot of that those polymeric compounds from the wheat straw. On the bacterial side of things, again, we get this rapid succession in phase one. And then we get this stable community in phase two and three, where these organisms work together to further break down the cellulose as well. So these organisms work in tandem with the Mycothermus. So Pseudoxanthomonas has been found in many cellulose degrading consortia. It produces beta-glucosidase, which breaks down cellobiose, a disaccharide of glucose. But this taxon itself has not been known to produce any cellulose degrading enzymes. Staurodobacter has also been found in lignocellulose degrading consortia. However, I haven't been able to isolate a Staurodobacter organism, so I won't really be talking about this organism much further. And finally, Chitinophagaceae, which is a family of known bacterial chitin degraders. For this particular data set, this taxon is a novel chitin degrading bacterium that I've managed to isolate, and I will talk about this a little bit more later on. So that was the profile of at one compost yard, and I wanted to see if we get a similar profile across at that stage across multiple compost yards in Australia. And we do get a little bit of consistency in diversity across different compost yards. So the ones that I'll point out are Chilatovorans and Chilatococcus, which can grow on a minimal medium using cellobiose, again, a disaccharide of glucose as its sole carbon source. And it's also been proposed that it aids in lignin breakdown. However, this hasn't really been confirmed in literature. And again, we see Pseudoxanthomonas, which is our dominant taxon in most of our yards and takes up a relative abundance of about 30% in some of them. And this also mostly consists of one species, Pseudoxanthomonas taiwanensis. So knowing that our profile looks not quite the same, but a little bit similar, how do these taxa interact with each other? So focusing on Mycothermus and Pseudoxanthomonas, what I had to do was isolate as many Mycothermus strains as I could get my hands on with the compost that I had. So I managed to isolate about 10 different Mycothermus organisms or Mycothermus strains from the different compost yards. And Sanger sequencing confirmed that all of these strains are Mycothermus. And then I used a DNA fingerprinting method called box PCR to confirm that these strains are all identical. So across our 5 different compost yards in Australia, all of our Mycothermus strains are exactly the same. Cool. Then I managed to isolate 3 different species of Pseudoxanthomonas taiwanensis. And then here I have them grown on plate in co-culture with Mycothermus. So here the bacteria have been streaked onto the plate. They've been grown to stationary phase. And then the fungus is inoculated in the middle and allowed to grow over the plates. And as you can see here, the Mycothermus growth is not halted by Pseudoxanthomonas taiwanensis or Pseudoxanthomonas coriensis. However, there is a bit of an inhibitory effect from Pseudoxanthomonas suinensis. Following on from that chitin-degrading bacterium that I've managed to isolate, which is called Microborax composti, I did the same method where I've grown it on the plate to stationary phase. And then as you can see here, it does release a sort of inhibitory compound that blocks or that stops the growth of the Mycothermus. Microborax grows at a very similar temperature to Mycothermus. It grows at about 45 to 50 degrees, which is its optimal temperature. And also at that phase 2, phase 3 stage. These plates only look at how they interact with each other once one has already been fully grown. And I wanted to see what would happen when both are growing at the same time, because usually that's what's going to be happening in our compost system. So what I did was overlay a nylon mesh that's inoculated with Mycothermus on some glass beads that's been sitting in some growth media. That Mycothermus is grown for about 4 days at 50 degrees, and then I'll inoculate the growth medium with a bacterial culture, and then further grow that for another 5 days or until the bacterial culture has reached stationary phase. And what I saw was that when the Pseudoxanthomonas taiwanensis was inoculated into that broth, the growth of Mycothermus was not affected as much, compared to the other 2 species of Pseudoxanthomonas coriensis and suinensis really halted the growth of the Mycothermus once it was inoculated into the broth. On the bacterial growth, so here I have them in OD, in the presence of the fungus, Mycothermus, all 3 species of Pseudoxanthomonas grew really, really well. However, without the Pseudoxanthomonas there, it didn't grow very well at all outside. So that leads me to believe that the fungus was releasing a compound or some sort of nutrient that boosts the growth of the Pseudoxanthomonas. With our chitin-degrading organism, here the bacterium grew better in the presence of Mycothermus. However, the fungus with the bacterium in there did not grow very well. In fact, it actually started decreasing in radial growth. So, and that was actually because the Microborax is using the chitin in the fungal cell wall of Mycothermus. And I actually was able to see that under the microscope. So here I have Mycothermus that have been stained with Congo red, which binds to chitin. And you'll see that the fungal cell wall of Mycothermus is completely intact, compared to when the Mycothermus was incubated with the Microborax. Here, the fungal cell wall is almost fully degraded, and it was actually quite fragile, where it was very difficult to get a full piece to see under the microscope, and it's also actually flaking off quite a bit. So we looked at the interaction between Mycothermus, Pseudoxanthomonas, and Microborax, but we have a whole plethora of other organisms here that are available to use and are there. So we've managed to isolate a Calatococcus strain called Calatococcus composti. And we wanted to see how that would interact with Microthermus, as well as what interaction that has with Agaricus. So unfortunately, growing Agaricus in a lab setting is a little bit difficult. It doesn't grow very well on lab media, but it grows very, very well on compost. So what we did was we got some test tubes, filled the bottom with some Agaricus spawn, filled the tubes up with our N phase 2 compost that's been inoculated with an inoculum, and then it grows up the tube for about 13 days till we get complete colonization by the Agaricus, and we call these race tubes. So a previous student in our lab did a whole bunch of, started off with trying to see if this adding extra microbes into the compost was just extra food for the Agaricus. So here she's inoculated Pseudoxanthomonas taiwanensis and Calatococcus into the compost, sterilized the compost by autoclaving, and then inoculated it with a Microthermus spore suspension, because we know that Microthermus does boost the growth of the Agaricus. She's done the same here, where she's sterilized it but not added anything, and then done the inoculation with the Microthermus, and so on and so forth. And then her control was just not adding anything at all and letting it grow. And what she found was that Microthermus does boost the growth of Agaricus mycelium, so great, that was confirmed. However, compost bacteria do have a positive effect on the growth rate of Agaricus. So having just the Microthermus there alone wasn't quite enough, and we get a better growth with bacteria there as well. Adding the bacteria and then sterilizing the compost and not adding anything post-sterilization actually caused the Agaricus to stop growing at all. And after, I think it was 6 days, it wouldn't continue growing. So great, now we need to figure out which microbes are beneficial. So she tried looking at only Pseudoxanthomonas or Calatococcus or Microthermus in the compost alone, a combination of the bacteria in the compost, or a combination of the bacteria plus Microthermus together. And she inoculated the compost where the bacterial cells were all grown to stationary phase, and inoculated it at 5 × 10⁷ cells per gram of compost. So quite a high concentration of cells. And what she found was that all of the combinations improved the growth of compared to the control. However, just adding Taiwanensis alone, the Agaricus growth was boosted the most. So that led me to believe that inoculum concentration, so previously we knew that in co-culture, or the Microthermus plus all the bacteria together gives a good growth. Maybe it's the inoculum concentration that needs to be optimized. So I focused particularly on just this organism, just this inoculum concentration, and did a serial dilution across all of them. And I found that too high of an inoculum concentration slows the Agaricus mycelium growth rate, and that our race tubes with the fastest growth rate also shortens the phase 3 time, so the time that it takes for our Agaricus to proliferate through the compost. So as I said earlier, a typical phase 3 process takes about 15 to 16 days. With the optimum inoculum added in, it only took about 13 days. So overall, the interaction between Pseudoxanthomonas taiwanensis and Microthermus are compatible in mushroom compost. Adding a consortium of compost microorganisms may be more than just extra food for the Agaricus, some sort of interaction going on between the two that I'm still working on trying to confirm. And also it will aid in shortening that composting time, which means that we can get more mushrooms on the table faster, hopefully. And that microbial composting may be involved in nutrient cycling and also feeding our mushrooms by releasing some carbon and nitrogen for the mushrooms to use. So current work that's going on. So I've taken those optimum inoculum concentrations and I'm trying that in an actual growth trial. So I've inoculated our compost with those concentrations, and that's growing at the mushroom unit on Darlington side. As Justin said earlier, we get these growth trials going, and then we have an excess of mushrooms that we end up giving away. So if you find yourself in Lee's during one of those growth times, definitely grab yourself a bag of mushrooms. It would also be good to see what other bacterial associations we can have with microthermus and that effect it has on the agaricus. And also how the growth of Microborax composti affects Agaricus bisporus. So again, running it through a race tube to see if it slows or increases the growth rate of Agaricus. I'd like to thank my boss and also Tina, who has helped me a lot with all of the work. The Curtis Lab team, Phil, Shiva, Rebecca, Margot, as well as Maggie, who did some of the bacterial isolations and purification, as well as all of the mushroom composters who were able to provide samples for this project to happen. Thank you, and I'll be happy to take any questions.
Thank you, Meghann. That's extremely interesting to hear about your work. I actually wasn't aware of how much work was going on with the mushroom industry at University of Sydney. So that's been cool. And it's great to hear about all the different techniques that you're using, and thanks for sharing your results. I'm sure there will be lots of questions directly related to what you've spoken about, but how also some of your techniques could be applied in different settings.
Yes.
But before we go on to questions, I think it'd be great, especially for people who are not in the University of Sydney and are watching this on YouTube later, if you could just, in very broad strokes terms, explain some of the work of the Curtis Lab and how you interact with the mushroom industry. And also for interest, how did your background lead you into that position?
I'll start with answering the second question first. So how did I end up here in the mushroom lab? Well, when I started my university career, actually when I started my entire education career, I wanted to be a vet. However, I ended up doing a few vet units and found that it was not my calling. And I went, oh, okay, now what do I want to do? Then I had a lecture from Michael about how to grow mushrooms and I was sold. I was like, if I'm doing a science, it has to be edible. It's mushrooms. So I started my microbiology path down that way. And then I did a little mini project with Michael in third year, looking at pathogens on mushrooms and their persistence, essentially. And then from there, it went into looking at the nitrogen cycle during cropping and all of that stuff. So I've done a lot of molecular work and biochemistry work in that sense, in honors. And then I went away to the industry, learned how to grow mushrooms, was picking mushrooms for a couple of mushroom farms as well, and then came back and was like, oh, I want to do some more research. I want to help the industry out a little bit more, see what I can do for them. So Michael, being the only person I know that was working on mushroom research at the time, and still is basically the only researcher working on mushroom research, I asked him if we had a project or we could do a project together. So he took me on, and 5-ish years later, I finished my PhD with mushrooms. How do we keep in contact with the industry? So every year when we get new students, we tend to try and take them out to the mushroom farm. So our closest farms that we have at the moment in New South Wales are in Windsor and Mulgrave. So it's about an hour drive from Sydney, and we do keep a close relationship with them as much as we can. Every second year, we have a mushroom conference with the growers. So we do try to do a talk for that conference every time the conference is on. Last year, Michael and I were kind of fighting over who was doing the presentation. I did the presentation in 2018 when he went on sabbatical, so I was like, maybe you do it this year. So yeah.
Okay, cool, great. And what's the scope of the rest of the work in your group?
In our lab,
yeah.
it varies. So a lot of mine is looking at still more focusing on nitrogen transformation, how that moves throughout the composting system, as well as a lot of the ecology that happens. So setting up race tubes and doing all of that. We have a PhD student who just finished her honours with us last year, who's now working on bacteriophage, which is bacterial viruses. What else do we have? We have another student in our lab who's going to be looking at the microbial community in phase 1 and taking a much closer look at what happens at that stage. How rapid is that succession occurring? What else do we have?
That's already a lot.
It's a lot. We have a pretty big team at the moment.
That's amazing. Thanks for sharing. Okay, so we have a first question. No, we don't. We have somebody who has to leave. Bye, Meghann. Meghann. But do we have any questions from anybody who's online now? I see Julie first is unmuted, and then Ruth.
So thanks, Meghann. That was a really fascinating talk. And look, I know you're trying to find, positive interactions between these, bacteria and fungi, so that you can promote, the growth of mushrooms. But I'm really interested in some of the negative ones you found, where I'm wondering whether you might have discovered a new antimicrobial. And I found that really exciting. I mean, it looked like your fungi were dying.
Yeah.
Because they weren't being stained with that Congo red.
Yeah.
And so, I just wondered if you're sort of interested in side projects where, you can kind of, take, like when you grow those plates, can you take some of that area or that zone of inhibition and send them off to a chemist or something to find out what they are?
Yeah, so I did do a little quick test to see, I did a chitinase test, so it was just a colorimetric chitinase test, and did confirm that it is producing chitinase, so it is breaking down. It is. Yeah. Okay. How do I go about purifying that? I am not sure.
I mean, I guess if it's a chitinase, sorry, I probably missed that. I got really excited thinking, wow, I'm fighting something else. But yeah, if it is a chitinase, then I guess that's a sort of I'm not sure whether we can exploit those for.
I'm not sure either, but it would be good to see if we could. So I'm currently looking at inoculating that in with the microthermos in the compost.
Mm-hmm.
So does it break down the microthermos and then therefore produce more food for the agaricus, as well as if I have it just alone in the compost, is it going to have a detrimental effect on the agaricus growth? The difference between the microthermos and the agaricus is that agaricus grows at a much lower temperature compared to the microborax, which grows at 45 optimally. And doesn't grow below 30. So I don't know if it will have an effect on the agaricus. Having said that, it might if it's got a food source to go with.
Mm-hmm.
Yeah.
No, it's good though, because you've got like a nice. I mean, I think that's what people are doing these days. Is they're looking at soil microbes and trying to find new antifungals. So you're in the perfect position to do that as a little side side job.
Maybe I'll team up with Aiden
Thank
in
you.
Dee's group.
Excellent side hustle. Ruth, your question?
Yes, thanks, Justin, and thanks, Meghann, for a fantastic talk. Unlike you, I did get stuck in vet science. I was really interested from the very start of your talk, where you talked about chicken manure as one of the sources for your mushroom compost. And in the animal industries, manure is often seen as a waste product rather than a resource. So if you can turn that waste into the resource that it really is in terms of nutrients, that would be fantastic. But I don't think we are really connecting the source of animal manure to the use for mushroom growing or other constructive purposes yet. So I wonder whether you or anyone else had looked at the role of the chicken manure in that compost mix. And I'm thinking specifically about things like different diets that chickens could be on, which will, of course, affect the composition of that manure. And then also different bedding materials that we're using in poultry sheds. And now I'm 100 meters down the road from a poultry shed. So maybe we need to do an exchange visit. I'll come and see your mushrooms and you'll come and see my chickens, and then we'll cook up a storm at the end.
Absolutely.
But yeah, can you talk a little bit about the composition of chicken manure, both in terms of the chicken's diet and in terms of the bedding that the chickens they're on, and how that might impact on your compost composition and productivity?
Yeah, so compost, so the chicken manure in the composting system is the primary nitrogen source. Having said that, again, with the changing diet and basically the nitrogen conversion in chicken feed being so much better now than it was before, a lot of that nitrogen it doesn't end up in that manure. It ends up in the chicken, which is where you want it to be, which isn't so good for our composters, because that means there's a decrease in nitrogen content in that manure. And then our composters have to use more manure in order to meet those carbon nitrogen ratios that they need for the composting to happen. So they often substitute a lot of that nitrogen in with other waste products. So they use a lot of feather meal or cottonseed meal as well, canola meal as well they've used. But yeah, the diet of the chickens I'm not so aware about, other than I know it is causing a decrease in nitrogen content in the manure.
Yeah, and Justin, maybe I need to follow up with Meghann offline if this is being recorded for a broader audience. But Meghann, we're specifically looking at diets of poultry to do exactly what you said, reduce the nitrogen content in their excreta and get it into the birds to make the poultry production more sustainable. We're actually potentially also competing with you for canola meal as a nutrient.
Oh no.
But we're running lots of nutrition trials, so we could actually see how mushrooms grow on poultry manure from different diets with different compositions, and we'll be measuring water content and nitrogen content. So maybe it's a conversation to take offline.
Yeah.
But
absolutely,
certainly opportunity to discover a whole new world together.
Yeah, definitely.
That's we're talking about integrated farming solutions, and how that might affect, I guess, the considerations that go into the chicken feed if you actually wanted a high nitrogen product as an output, as well as getting more nitrogen into your chickens. It's really interesting.
Circularizing the economy is a big thing now.
Yeah, well, that's exactly what we're talking about for the animal farms that the university owns. So I'd love to continue that conversation.
Yeah.
Amazing. This is why we love Fungisphere meetings, bring everybody together from such different angles. And are there any other questions from anybody online? Dee, I see you wave. Unmute.
I'll ask a question, even though I could just ask you in the office out there. I was really interested to see penicillium, kind of that little peak of penicillium.
Yeah.
In phase 1, because it's not a thermophile.
No.
it surprised me because the only thermophilic penicillium relative is Telleromyces, which is,
Yeah.
So have you got an explanation for that?
I have a feeling, so that data is a little bit old. It was from about 2015, and that was sort of before, actually, I think it was, no, that actually was from 2012. So that was before a lot of the fungi got reclassified, and
Yeah.
it may have been reclassified as something else. I have to look at the data again and update it with the new database to see if it actually is.
With DNA data, or was it culturing data?
DNA, yeah, it was all diversity profiling. Yeah. But
Okay.
that wasn't done by me. That was done by a predecessor in our lab.
Okay, I would be a little worried about a thermophilic penicillium.
Penicillium, yeah. Yeah.
It's
No,
just
I don't I suspect it actually got reclassified as something else.
Right.
Yeah.
Yeah, and I was equally surprised you didn't see Aspergillus because Fumigatus is like the compost organism. So
Yeah, so it is there, it's just not very high.
okay.
It's less than 1% in the compost. So
That's surprising. I would sort of would
yeah. Yeah, I would have thought it would have been a lot higher as well, but no, it didn't actually come up that high. Yeah. Yeah.
Also, just a follow-up from Ruth's question about the chicken stuff. They use a lot of antifungals in chickens, right? In terms of treating their feed and treating them as well. Is that likely to be an issue with then growing fungi?
We do not think so. I mean, it certainly hasn't really impacted the mycothermus. Um,
Okay.
I think the general consensus is that because it's composted, so it ends up
Yeah,
being inactivated. Yeah.
that's true.
Yeah.
It would be interesting to know if there is any trace residues of azoles or DMIs for agricultural use in the compost, and whether
Yeah.
I've actually no idea whether button mushrooms are susceptible to azoles.
Just and indeed it's a very good point, because one of the reasons we're doing our current work is that a lot of the poultry diet ingredients are imported from overseas, which is not good for the carbon footprint of chicken production, but also not for the chemical quality of the ingredients, because controls on use of antifungals and antibacterials and antiprotozoals are not as strict elsewhere as they are here. So it's certainly something we could have a look at. I'm not sure we make friends in the poultry industry, and as Meghann will know from her work with the mushroom industry friends are important, because ultimately they are our key stakeholder, and any recommendations would hopefully be implemented by them. So interesting terrain to explore.
Yes.
Thank you for joining us. I hope you've enjoyed today's talk from the Fungisphere webinar series, and we look forward to seeing you next time.